Hi, everyone. This is Anna Roberts Pilgrim, and we're bringing another edition of the Office of Cancer Clinical Proteomics webinar series. Today, Dr. Eric Genig will be presenting on the LinkedOmics software, first developed in the lab of Bing Zhang at Baylor Institute with Eric, and he will be walking through how to use the program and new features of the program. We're really excited to have him here with us. So we'll get started on our webinar, and we will do our basic intro slides, and then I will turn the presentation over to Dr. Genig. Thank you.
Thank you for joining the NCI's Office of Cancer Clinical Proteomic Research webinar series. Part of NCI's Cancer Continuum, OCCPR mission is to improve prevention, early detection, diagnosis, and treatment of cancer by enhancing the understanding of the underlying molecular mechanisms while advancing proteome and proteogenome science and technology development. Through the integration of genomics and transcriptomics with proteomics, also known as comprehensive proteogenomics, OCCPR's aim is to advance basic, translational, and clinical research through OCCPR-supported programs. This webinar series hopes to educate you to all the resources OCCPR and its researchers have to offer.
Awesome. Now we'll turn the meeting over to Dr. Genig. And so, Dr. Genig, I will stop sharing my screen.
Okay.
And I will pass you the ball. And there you go.
One second. Sorry, I at least have trouble finding what the share part is. Option is for.
So it's usually.
Okay, I got it. You can see it now?
Yes, perfect.
Okay, great. The slideshow started. So as Anna, I'm a research associate in Dr. Bing Zhang's lab at Baylor College of Medicine. And the Zhang lab is one of the proteogenomics data analysis centers for the Clinical Proteomics Tumor Analysis Consortium, or CPTAC. One of the primary objectives of CPTAC is to enable researchers to analyze the proteogenomic data sets from clinical cohorts that we are generating. And today I will show you how to use a tool that our lab has developed so that you can analyze the multi-omics data sets that we're generating through CPTAC yourself. LinkedOmics is a web tool that can be accessed at the linkedomics.org website. And it's a tool for exploring links or associations between a query feature or attribute in one data set and all of the features of another data set. LinkedOmics was primarily developed by Suhas Vasakar, a former research associate in the lab, and it consists of 4 components. The first component is essentially a database that consolidates all of the data from TCGA and CPTAC. The Cancer Genome Atlas, or TCGA, was a multi-institutional effort spearheaded by the NIH between 2006 and 2017 to generate a large collection of multi-omics data from clinical cancer cohorts. Altogether, they collected 7 different types of omics data from 30 different types of cancer, spanning approximately 11,000 patients. LinkedOmics contains the process data from all of these omics data sets. What I want to draw your attention to is right now is one of the data types here, RPPA. This was TCGA's effort at collecting proteomics data. It involved protein arrays. RPPA stands for reverse phase protein arrays, which only allows you to actually measure a very limited number of proteins at the same time. So typically we'd see less than 200 proteins with protein measurements in the TCGA data. However, mass spectroscopy allows us to quantify thousands of proteins from the same sample, in addition to measure post-translational modifications such as phosphorylation or acetylation on these proteins. Therefore, the NIH has organized a second multi-institutional project, the Clinical Proteomics Tumor Analysis Consortium, PTAC, mass data. The first round of CPTAC involved involved based proteomics analysis on residual material left over from three TCGA cohorts, the colorectal, breast, and ovarian cancer cohorts. Since then, however, CPTAC has generated data, or analyzed and published multi-omics data for 3 additional cancer cohorts: colon cancer, endometrial, and clear cell renal carcinoma. In addition, manuscripts have been submitted for 3 more cancer cohorts: lung adenocarcinoma, breast cancer, and head and neck cancer. So all of this data from these cancer cohorts will be available soon. And as soon as they're published, they will be available from LinkedOmics as well as the CPTAC data portal. The remaining components of LinkedOmics, the link finder, link interpreter, link compare modules, allow users to analyze links within these data sets and between these data sets. The first module in LinkedOmics is link finder, which allows you to perform statistical tests to evaluate the associations or links between a query attribute in one of your data sets and all of the attributes in a target data set of your choice. So as shown in the table here, that the type of statistical test that Link Finder performs will depend on the type of data you're using in your search. For example, if your query attribute is a binary attribute, such as whether or not a gene is mutated in a given sample, then Link Finder will allow you to perform either a t-test or a Wilcoxon test if the target data is continuous, or it will perform a Fisher's exact test if the target data is binary. So to expand upon this example further, say for example, you're interested in identifying proteins affected by mutation in the PIK3CA gene. Your query attribute would be a binary attribute of whether or not that gene is mutated, and the target attribute would be a continuous attribute, which is the expression level of all the proteins in the proteomics data set. So what Link Finder will do Link Finder will perform a t-test comparing the protein levels for the samples that have the PIK3CA mutation, that don't have the PIK3CA mutation. Each protein in the proteomics data set. And you can plot your protein of interest as a box plot as shown here. If your query attribute is a continuous attribute, such as the expression level of your favorite protein, Link Finder would allow you to perform Pearson or Spearman correlation tests if the target data is continuous, or a t-test or Wilcoxon rank sum test if the target data is binary. But if we were interested at the correlation of a particular protein, such as in this example here, ZEB1, with the microRNA level, with microRNAs from the microRNA data, you could perform Pearson correlation analysis for the correlation between ZEB1 and each of the microRNAs. And you can choose your favorite microRNA and plot the correlation results as a scatter plot. The second module is the link compare module. Link compare allows you to compare the results of multiple compatible link finder queries, enabling multi-omics analysis within the same cancer cohort or pan-cancer analysis across different cancer types. For example, we might be interested in looking at how well MIR200C correlates with RNA levels and how well those correlations line up with the MIR200C, the microRNA 200C correlation to the same protein. So this plot here that link compare enables shows the correlation statistics for MIR200C with all the genes at the RNA level, and then the corresponding correlation at the protein level on the y-axis. Another possibility is that we might be interested in determining which phosphorylation sites are affected by PI3 kinase mutations in both breast and lung cancer. And link compare would allow you to, so the way you'd set this up is two link finder queries, one for breast cancer and one for lung cancer. And then link compare would allow you to look at the overlap between these two queries. Another possibility is to perform meta-analysis on greater than two link finder queries. The final module in LinkedOmics is the link interpreter module. Link interpreter allows you to evaluate enrichment for gene sets using the results from either link finder or link compare. Specifically, the link interpreter module calls upon another tool our lab has developed, WebGestalt. Allow you to perform overrepresentation analysis for genes that show significant associations from your query, or to actually use the statistic from the query for all of the genes and perform gene set enrichment analysis. Enrichment analysis can be performed for any of the gene sets in the gene set databases shown here from gene ontology databases, pathway databases, and network databases. So now that I have shown you what you can do, what types of analysis that you can do with LinkedOmics, let's go through an example and see how to actually use it in a demonstration. For this demonstration, what we're going to do is look for proteins that are significantly associated with survival in the TCGA CP-TAC ovarian cancer cohort. And this actually comes from from figure 4 of the LinkedOmics paper. This is the figure from the LinkedOmics paper, and we're going to generate all these plots here. So in addition to looking for associations of proteins with survival, we're also going to look at associations with copy number at survival in order to use link compare to compare these two queries and see which genes have both copy number changes and protein levels that are significantly associated with survival. The idea being that copy number change might be driving the protein levels, which is what's important for survival. So let's go ahead and get started with the demo. So what we need to do first is go to the LinkedOmics website. If you just collect LinkedOmics or type LinkedOmics.org in the address bar, that will bring up this homepage. If you're going to be using LinkedOmics frequently, I would recommend that you register so you can save your searches and don't have to repeat them every time you want to come back to it. Everything that we're doing today, we can do as a guest. So if you want to follow along, you can enter as a guest. This is up the page for the first step. Any analysis in LinkedOmics will begin with a link finder query. So the query that we're going to do here is to look at the protein levels from the proteomics data that are associated with overall survival in the ovarian cancer cohort. The first step is to select the cancer cohort. This doesn't show everything. You could choose to show everything, but it's actually probably a little bit more convenient to search for it. And this is an autofill search. So you just need to type in a few characters like OV for ovarian, and it will bring up the only data set with OV in it, which is ovarian cancer. So we can just select that. The next step is to select the search data set. So the search, our query attribute is going to be overall survival. So our search attribute, our search data set is going to be clinical data. I think the easiest way to get to the clinical data is to go to this data type column and then click on the drop-down menu here. It'll bring up the different data types in this cohort. And here we can select clinical. We'll fill in with all of the data sets that have the data type clinical data. There's only one here, so we'll select that one. So there's an optional step that brings up, which is to select the sample data set. We're not going to use this today. If you are interested in doing a more focused analysis, you can always click on this and then select a clinical subset of interest that you want to focus on. We're going to use all of the samples here, so we're not going to do that. And the search attribute is going to be overall survival. That's the next step. We select that. And then our target data set, we're looking for proteins associated with survival. So our target data type is proteomics data. So we can select proteome from the data type pull-down menu. And there are two proteomics data sets. We're going to select the second one. And that brings us to the final step, which is to select the statistical method. For survival analysis is Cox regression analysis. So we'll just select that and submit the query. And then this brings up the results from your link finder query. This might take a while to do if you're doing a search for the very first time, but LinkedOmics does save the searches for a while. So if someone's already done the search, the chances are you might not have to wait. I've done this search already, so we don't have to wait. If we want to see the results, we just click on the view button here, and that brings up the results below. So there are two tabs here. The first tab is for the link finder results, and the second tab is for link interpreter. That tab is for sending the statistics from your link finder query to link interpreter to send enrichment analysis. So let's first focus on what's in the link finder tab. There are 4 items in here. The first is a table showing all of the statistics from your query. It has a list of the attributes, and then the second column contains the statistics. So in this case, since we did survival analysis, it's the log of the hazard ratio. Next column contains the p-value and the FDR corrected p-values in the third column. The second plot that is shown in the link finder tab, the right here, is a volcano plot on which the x-axis for your the x-axis plots the statistic from your test, and the y-axis plots the minus log 10 p-value. And each point is a specific feature from your analysis. So, for example, here, the strongest gene is SEC24C. That's going to be this plot here. You can click on interactive plot to actually bring up a plot in a separate window where you can click on points and see what they are. And then the final two panels in heat maps of the positively associated genes and negatively associated genes. So let's take a closer look at the top hit here. The SEC24C is the protein that shows significant association with survival. Its log hazard ratio is negative, which means that the hazard ratio is less than 1. That means that higher levels of expression of this protein are associated with a better survival because the hazard ratio is represents the associated with having an attribute. LinkedOmics uses high expression as the indicator for having a particular attribute compared to the samples that don't have that attribute, that have lower expression. We can click on view to see the Kaplan-Meier plot for SEC24C. So as I just mentioned, LinkedOmics will separate your samples by their level for the protein, SEC24C. And samples with expression above the median will be the samples for which you're gauging your hazard ratio. So those have actually better overall survival than samples that have lower levels of expression of SEC24C. What this means essentially is that there's low risk associated with having higher levels. So let's go ahead and try out the third module in LinkedOmics, Link Interpreter. So we're going to send the signed log p-values from our link finder query to Link Interpreter. So just click on the enrichment analysis. So as I mentioned before, we can do overrepresentation analysis of the significant genes or send the signed log p-values for all of the genes and do gene set enrichment analysis. Let's go ahead and do that. And let's take a look at pathway databases. Let's look at the pathways in the wiki pathway database and see if we see enrichment for proteins associated with survival in wiki pathways. So go ahead and click on that and start the search. This takes a couple of minutes to do in WebGestalt, but I have it's blocked by this. Okay, here we go. So basically, what you see is pathway enrichment. At the top here, this shows this shows the pathways that have enrichment for genes with positive signed log p-values from our link finder search. And most of these look like they're immune pathways. Let's scroll down and take a look. It also shows bar plots for pathways that are enriched for genes with negative signed log p-values from our Cox regression analysis. Let's take a closer look at an example of one of these. For example, I'm going to select the DNA mismatch repair. If we click on the bar, it will bring up the specific search results for that pathway. This includes an enrichment plot here showing the rank of the signed log p Cox regression analysis for the proteins in the pathway. So as you can see here, the higher rank means more positive signed log p-value from the regression analysis, whereas on this end you have the negative values. So you can see here there's an enrichment for proteins that have a negative signed log p-value from our association analysis. And actually the table below shows the statistics for these leading edge genes. For example, you see here that RFC5 has the most negative association with survival, followed by MLH1. So prior to this talk, I actually went and did a literature search for mismatch repair, the pathway that is highlighted here that comes out as being significantly enriched, and ovarian cancer, and found this paper entitled Prognostic Values of DNA Mismatch Repair Genes in Ovarian Cancer, Patients Treated with Platinum-Based Chemotherapy, which reports that high mRNA levels of MSH6, MLH1, and PMS2 were significantly associated with better overall survival for ovarian cancer patients, which is consistent to what we observe here. And we can go ahead and take a look at that specifically if we go back to our link finder query and search that table, the table of our statistics for MLH1. And go ahead and view the Kaplan-Meier plot for this. Sorry, it's taking a few seconds to load this. So, oh, there it is. Okay, sorry. So we can see again that higher levels of MLH1, the red curve here, are associated with better overall survival than lower levels of MLH1. So the Cox hazard ratio here is going to be less than 1. So the sign log p-value was negative. So this is all very consistent with that previous study and with the results that we see for the pathway in general. However, as I mentioned at the beginning of the demo, what we're interested in doing is looking at proteins that are associated with overall survival, for which those protein levels likely result from changes in copy number. So we'll go ahead and set up a new analysis where we look at associations of copy number changes with overall survival in this same cohort. So again, we select the ovarian cancer cohort, and we're looking at overall survival. So the search data is clinical data, and the search attribute, or the query attribute, is going to be overall survival. But this time the target data will be copy number data. So we're going to select SCN, and specifically we want the gene level copy number changes, because we want to see the genes that are in copy when we look at protein and copy number associations. And we'll use the continuous data. So we're going to select this data set here. And again, since it's a survival analysis, we do Cox progression analysis, and brings up the results here. So in the interest of time, we're not going to go into these results. We're going to skip ahead to doing the link compare analysis. So we can do link compare analysis of multiple link finder queries by selecting the boxes for those queries. So this is the copy number association analysis that we just set up. Go ahead and click that box and then click on OK. And then here is the proteomics search that we just went through before we did the copy number search. So we're going to have to select that too. So we've selected these two link finder queries, and now we're going to compare them using the link compare module. And that brings up a table, takes a second to load, showing the statistics for the each of these link finder searches. So basically, it shows all of the common genes between the two queries. And the first three columns show the statistic for the first query, which is the copy number analysis, and the statistics for the second query, which is the proteomics analysis we did first. And then the final three columns show summary statistics that combine from each of the link queries. What we're going to do here is actually take closer look at the Venn plots, because what we're interested in were proteins that also had copy number changes that were associated with overall survival. So we get two plots, two Venn diagrams here, one for the positively correlated genes. So these are the genes that have positive associations with survival and negative associations with survival. So we'll go ahead and click Open Window in New Page for the positive associations. And so the genes that have higher protein levels that are associated with, I believe, worse prognosis and higher levels of gene amplification associated with worse prognosis will actually overlap between our copy number search and the proteomics search. So we can go ahead and see what those are by clicking on the overlap data box here. And this shows a table for all of the genes that are overlapping between these two queries. And the top hit here is actinin 4. We can click this to search the gene in NCBI if you want to learn more about it. But let's instead go back to our original search and take a look at it in the proteomics data. So go ahead and unselect our link compare selections and then select the original query, which is the proteomics search. And let's go ahead and search for the gene actinin 4 and take a look at the Kaplan-Meier plot. Okay, sorry, that's taking a second. Let's just go back to the PowerPoint presentation. Oh, it was there. Yeah, here it is. And actually, we'll just look at it in the PowerPoint presentation since it's there as well. So we actually generated all of these plots except this plot here, which is the core copy number Kaplan-Meier plot for actinin 4. But as you can see here, higher levels of actinin 4, which is the red bar here, are associated with worse prognosis. Same for the copy number data. So our findings are actually consistent with some previously published research that found that actinin 4 gene application in ovarian cancer results in poor prognosis and chemoresistance. Okay, so I just want to summarize by reminding you that LinkedOmics is a database that contains multi-omics data sets from TCGA and CPTAC. And it also for analyzing links or associations within these data sets and between these data sets. And I want to thank the members of the Zhang Lab, particularly Bing Zhang, our PI, and then Suhas Vasakar for developing LinkedOmics when he was in the lab, and now Yujing Lao for maintaining the website currently. And I want to thank you for your attention, and I'll be happy to answer any questions.
Thank you, Dr. Jay. That was very interesting, very nice. So we have a couple of questions. Well, first I'd like to say some of the questions are whether or not the webinar will be available. It will be available. It's going to be recorded and it'll be available on the OCCPR website. We'll send out an email blast to let you know when that will be available. Another question, how long does the LinkedOmics save the searches when you do your initial searches? How long does the LinkedOmics database save that?
Yeah, that's a good question that I unfortunately do not know the answer to. I had actually I had thought it was forever, but then I found when I was preparing for this demo that it had to redo some of the searches that I had done before. So I ask Yuxing. I think it may just be like a first come first serve sort of thing where like you have to make space for when new searches are performed, you have to make space for those searches by eliminating the oldest search.
Right, okay, sounds good. Here's another question. It says, not clear whether positive or negative log values associates with good or poor outcome. Is it possible to have an annotation in the tool that helps explain that?
Yeah, I think that will be good. Sorry, here. I'm gonna make a note of this. I think I'm gonna ask you about whether or not we can clarify that.
Sounds good.
Because I guess what it really it really right now requires you to understand the statistical test, Cox regression analysis, and what a hazard ratio means. So that's not very useful for someone an ordinary biologist.
Yeah, that would be helpful. Okay, so here's another question. Is it possible for us to upload our own unpublished multiomics data?
Yes, that is possible. You have to contact. I think you'd probably have to contact Bing to arrange that.
I think we have a couple more questions. Here's one. When can we expect the data for gliomas to be integrated?
Is that published?
I'm not sure it is quite
I
sure.
don't think that's published yet. We typically don't. We typically make the data available once the paper is published. So if we go back to this slide here, the all of this data at the top is available right now. The clear cell renal carcinoma has been published, but we just haven't entered it yet into the database. These other three data sets where the manuscripts have been submitted are actually already in there. They're just restricted access right now. So I guess it would really depend on when that gets published, because even if if it's available at the CPTAC data portal before publication, because I know there's an embargo that once that's up, even if the paper's not published, the data is available. We typically wait until the paper is published before we make it accessible in LinkedOmics.
Answers that question. That's good. Here's another question. Is there an option to build a local instance of LinkedOmics to analyze and view our internal data sets?
Beyond, I don't know the answer to that. I think we would have to ask Suhas. So right now, I think the way it's set up is this is all running over the web server. So like the previous question asked about uploading your own data to LinkedOmics. I think that actually would be pretty easy to do. And that's, I think in the paper we state that we're willing to host that data if you want to upload your own data and then use the LinkedOmics web server. But as far as building a local version, I don't know actually.
Sounds a little like a lot of work.
Yeah.
Okay, I think we have one last question. Is it possible to limit the test to, for example, 2 user-defined subsets of samples?
Yes. Actually, that's I guess maybe I didn't make that clear, but I did point that out when we were doing the search. Let's quickly just sort of repeat it, and I can show you where to do. Actually, we're gonna search a different data set because there's better examples in the breast cancer data set. So let's select the breast cancer data set here, or the cancer cohort. I don't think it matters now what the search data set data type is, what search data set you're using. It's just that the step 2B after you select the search data set allows you to select sample data sets. So here we can click And you can subset the samples based off of clinical attributes. For example, if we want to focus specifically on HER2 positive samples, we can select this button here, and this will subset your SERT. So you're not using all of the breast cancer samples, you're just using the HER2 positive samples for your analysis.
That sounds good. Okay, let's squeeze
But
one more. Sorry, go ahead.
so I was just saying, but so in order to subset data, that it has to be in the clinical data file. So if you have an attribute that is not there, I guess what you do, if you have annotations for all the samples from the cohort with that attribute, is that you could ask us to upload a clinical data file that has that attribute.
Okay. I'm going to squeeze one more question in, one final question. Would the program be able to integrate immunopeptidomics, immunopeptidomics data in there?
Yes, I think that should be possible, and I think that may be something that we were going to do in the future. Um, I think it would be treated, it could either be treated at the gene level or at the peptide level. Level would be whether or not, actually, I don't know about the gene level. I think at the peptide level, we could do it the same way we handle our site level data. So for the site level data, we have, instead of using the genes as the attribute, we use the specific phosphosites. So you could use immunopeptides as the attribute and add that to the data set. It does limit some of the downstream analysis. You can't do the gene-based downstream analysis, such as the gene set enrichment analysis.
Oh, well, thank you so much, Dr. Janek. That was excellent, and I think it was very informative to walk through everything. Wonderful job.
Thank you.
I will take over the screen now and do our closing remarks. Thank you, everyone, for attending our LinkedOmics webinar. It was a wonderful presentation. It will be made available on our OCCPR website in the future, in about 2 to 3 weeks, where you can access it and be able to re-watch the video and see all the things you might have missed.
So
Sorry.
I'm going to interrupt just for a second. So we also would like to post a link to that on the LinkedOmics website. We try to post links to all of the talks we do and the tutorials that we give, and we have written tutorials available there as well. So there are lots of resources available to get more help in your analysis.
That's great. And I think we'll put links to it as well on our website so that people who watch the video, the webinar, can also link to that. That would be a wonderful idea. I will. Again, thank you everyone for joining. Say good day, and I will put up the exit slides information for you. Thank you again.
Thank you for joining the Office of Cancer Clinical Proteomics Research webinar series. Please visit our online resources included here and take a minute to answer our online survey. The Office of Cancer Clinical Proteomic Research is part of the U.S. Department of Health and Human Services at the National Institutes of Health, National Cancer Institute. For more information about OCCPR, please visit www.proteomicscancer.gov. For general information about cancer or clinical trials, please call 1-800-4-CANCER.